Hello, good morning to all. Today we are going to discuss about immune system, and that is cellular and humoral components. What is immunology? Nowadays we are saying that immunology immunity is discussed so much in connection with the COVID-19. So we must understand what is immunology and what is immunity. Immunology, that is the study of defense mechanism exerted by the body to recognize non-self or foreign matter. Example, microorganism and non-microbial foreign substances, maybe bacteria, maybe virus, and maybe anything, protein or carbohydrate and polysaccharide and nucleic acid, like that. And the body immune system destroys or make them harmless. And what is immunity? Immunity is the resistance exerted by the host towards injury caused by microorganisms or their products. The complete response of the body against any foreign agent, that is known as immunity. And that is of two types, that is innate immunity and acquired. Innate immunity is natural, which comes to you naturally, and acquired immunity is adaptive. With the when we get infection, then we develop that type of immunity. The evolution of immunity non-specific, innate immunity is non-specific, and that is immediate onset. And acquired immunity is specific, and that is of delay onset. And acquired immunity is of two types, that is humoral and cellular. And humoral, that is antibodies production, and in cellular response, that is T cell activation, that may be T cell activation. And what is acquired immunity? Resistance to infections, specific response to any particular disease, and acquired by exposure to an agent during his or her life. And it is highly specific response and having execute memory too. And that is of two types, humoral and cell mediated. Humoral response, you see B cell is activated and antigen attaches to antigen, and it converts or activated into plasma cells, and that produces your antigen. And in the cellular response, the T cell is activated. There is TCR, that is T cell receptors, and it is divided into T helper cell and T cytotoxic cells. And the T helper cells again activate the B lymphocytes and the T cytotoxic cells, and then it is converted to cytotoxic lymphocytes. And what is an antigen? Is the substance which enters our body, evokes a specific immune response, either by producing a specific antibody or by producing specifically sensitized T cells, or both. Any foreign substance may act as an antigen. What is antigen epitope? Entire antigen doesn't react but have special discrete sites which are called epitopes, and these epitopes are recognized by T cells and B cells. And what is antigen presenting cells? These are macrophages, dendritic cells, and B lymphocytes. And what is the function of antigen presenting cells? That is processing of antigen and its presentation to respective lymphocytes, that may be T lymphocytes or that may be B lymphocytes. There is antigen, and here is the antigen presenting cell. Here is the TH helper, that is type 1 helper lymphocytes. It produces lymphokines that activates the cells, and the lymphocyte converts into monocytes convert into macrophages, and here this T lymphocytes converts into or activated into cytotoxic T cells, and that leads to lysis of infected cells. Processing of antigen. What happens after entering the antigen in our body? Degradation of antigenic protein into small peptides, and that again associated with MHC molecules. And how it is presented? So these antigenic peptides expressed on the membrane along with appropriate MHC. Maybe there are different types of MHC molecules, and then presented to Th4 and Th8 cells. And this process is very meticulously controlled at several checkpoints to avoid non-specific stimulation of T and B cells that leads to autoimmunity and auto destruction of the cells. What are the organs of adaptive immune system? Primary or central lymphoid organs, that is bone marrow and thymus. Secondary peripheral lymphoid organs, those are lymph nodes, spleen and mucosa associated with lymphoid tissue. That means in respiratory system and in GI system, Peyer's patches in intestine, like that. The molecules involved in adaptive immune response. These are MHC molecules, membrane bound receptors, cytokines and cytokine receptors, chemokines and chemokine receptors. MHC molecule initiate the immune response by presenting the antigen peptide to TCR and BCR, that is T cell receptors and B cell receptors. Membrane bound receptors, this give first and second line signals for activation and bring about cell adhesion and cooperation. The cytokines are molecules that help the cells after recognition of antigen for clonal expansion and differentiation into effector cells. The chemokines and receptors help in cell trafficking and their migration and release through the capillary endothelium from the place of activation, maturation to the place of action, that is the site of infection. Major histocompatibility complex, that is MHC. MHC molecules, product of MHC genes, highly polygenic, that means large number of genes and same function, and polymorphic, a number of allelic variants of each gene. These are a special group of proteins occurring on the surface of T lymphocytes and known as MHC. MHC in humans are referred as human leukocyte antigen, that is HLA. No two persons other than identical twins have the same sets of MHC, so these are known as genetic markers of the biological cell. And these MHC molecules play crucial role in recognizing antigens on T cells. They are expressed on the cell membrane. MHC is of three types: class 1, class 2 MHC and class 3. Class 1 T cytotoxic cells, class 2 T helper cells, and class 3 is on self MHC molecules, that is the phenomena that is known as MHC restriction, only occurring when antigen is present on self MHC. Cytokine soluble mediators, low molecule weight proteins, glyco-proteins produced locally, activated by lymphocytes, macrophages, and act over a small range, involved in proliferation and differentiation, and leads to amplification